Section 5.3 looks at the life cycle of viruses. And as we look at the life cycle, we'll also look at what cytopathic effects are, and then look at some of the different types of infections that are caused. Alright, so looking at the life cycle of viruses in animal cells. Alright, so animal viruses, there are 6 major steps within this process. The first is going to be adsorption, right? Not absorption, but adsorption, when the virus locks on to the surface of the host cell. And so it's going to adsorb, and it's going to do this specifically through the interaction with that virus spike and a receptor on the host cell. Now, what we'll see shortly is that in figure or table 5.4, penetration and uncoating are grouped into one step. So after the cell locks onto, after the virion locks onto the host cell, it's going to penetrate the cell and enter the cell, the virion, and then it uncoats, and then it goes through the process of synthesizing new virions, assembling them, and then releasing them. Alright, the length of the replication cycle varies from 8 hours in, say, poliovirus to 36 hours in some herpes viruses. So adsorption, and we're going to go through these quickly because you can write this information down, and it's within your textbook. Adsorption is simply that attachment. So a virus can invade its host cell only through making an exact fit with a specific host molecule. So similar to a lock and key, it has to be the exact fit. And there's a host range, the limited range of cells that a virus can infect. So hepatitis B virus infects liver cells. Poliovirus infects the intestinal tract and then can move into nerve cells, but it doesn't affect the heart, doesn't affect the lungs. Rabies viruses, this one's interesting because it actually affects a variety of cells, and that's why it can do a lot of damage and have different ramifications within the body. If cells lack compatible virus receptors, then they're going to be resistant to that adsorption process and cannot be invaded by the virus. So what all this is called is tropism. Tropism is the specificity of viruses for certain tissues. And if there isn't the proper interaction between the virus and the host cell, then the virus is going to stay outside of the cell. And if it does that, it just gets washed away. It doesn't do anything. And while you may be exposed to the virus, there isn't enough there to actually cause you to have an infection. So this is what adsorption to the host cell looks like. Very similar to figure 5.4. Here you have the host cell membrane, and these little red pieces here are the receptor. And those are going to be a specific fit for this virus envelope spike. Sits right in there nicely, and that's going to actually signal the host cell membrane to pull that cell in, or to pull that virion in, right? That process of penetration. Here you have a different class of receptors. Alright, you can see the difference between them, and the capsid spikes are going to dock on to the surface of this host cell and be able to enter the cell. And what you want to keep in mind is that, well, this green ligand or protein spike locks onto this green pad on the host cell. It's not going to do anything against this red area, this red receptor on this other host cell. And likewise for this guy. All right, if this virion hopped over here and tried to get into this host cell, it's not going to do it by those green receptors. All right, so figure 5.4, you have 5 steps here, even though step 2 here has both the penetration and encoding. All right, so you have the adsorption, all right, the adhering of the virus to the host cell. You have penetration and encoding. And then you have synthesis, step 3, where all the material within the cell is going to be synthesized. 4, you have assembly. All right, now you have the new capsomeres, you have the new RNA. It could be DNA. And then, again, by some unknown direction or mechanism, you have assembly, where the capsomeres come together around the nucleic acid to create this virion. And then you have release. And in this case, you have release of an enveloped virus. The nucleocapsid is pushing through the plasma membrane, and when it does so, it takes some of that plasma membrane. Now, another thing that occurs with virus synthesis is you see these new spike proteins. All right, these are going to be created, synthesized on the interior of the cell, but similar to host cell proteins that get directed to come to the plasma membrane. All right, these guys also come to the plasma membrane, the new spikes. And so then when this virion buds out of the host cell through through exocytosis, it's going to have those spike proteins on its envelope. And those spike proteins are going to allow it to specifically bind to a new host cell. Alright, so the way a host virion can enter a host cell is through engulfment. Alright, what you see here, the entire cell is engulfed. The entire virion is engulfed. Or it can enter through membrane fusion. The membrane fusion is only going to occur with a virion that has an envelope, because the envelope is then simply going to bind to, via the spikes, bind to the plasma membrane of the host cell, and the membrane of the host cell and the virion are going to fuse together, and that's how the virus enters that cell. So in synthesis, you simply have replication and protein production, replication of the virion's nucleic acid, and protein production. You just simply have to replicate and synthesize new material for those new virions to be assembled. Alright, then finally, after you have that assembly process, you're going to have maturation and release of the enveloped virus. Alright, again, that release is going to occur through budding off of the plasma membrane. And you see examples on both sides of the slide, cartoon depiction, and then you have a electron micrograph picture of this process occurring. And what each shows you is just the process of this occurring, where you're going to have production, you're going to have assembly, and then you're going to have budding off. And in that process, you complete and create a fully mature virion. Alright, so real fast talking about viruses and cancer. It's been known for decades that viruses will cause cancer. And if a virus specifically causes cancer, it's called an oncogenic virus. It's estimated that about 20% of human cancers are actually caused by viruses. And what happens is this process of transformation. All right, transformation is the transforming of an individual, the transforming of a cell. And what happens when an oncogenic virus infects a host cell is it has a very negative effect. The oncogenic virus has a very negative effect on the host cell and transforms it into a cancerous cell. And what that means is there is an increased rate of growth. There can be alterations in chromosomes. There can be changes within the cell surface molecules. And what happens is this really is mutating the host cell. But under normal circumstances, when a host cell will mutate, a lot of times that signals the cell to die because it's messed up. But the presence of that oncogenic virus gives it the capacity to divide for an indefinite period of time. And so then that one cell, that rogue cell, that cancer cell, divides very quickly, producing more cancerous cells. So, with that idea of the increased rate of growth and the capacity to divide for an infinite period of time, that's how some tumors can not be present, day 1 and day 3, or day 4, day 7, be affecting cognitive function if we're talking about a brain tumor. And that unfortunately will happen. You have someone who seems to be normal, and then one day he or she forgets something, and the next day he or she forgets another thing, and then 2 weeks later there's something else added to the picture, and the next week something else occurs, and they go to the physician and they're told that person has a huge brain tumor, when a month ago they seemed completely fine. A month ago they probably still had the tumor, but it was still very small, that it wasn't putting pressure on anything or interfering with normal processes. Alright, so ways that viruses can cause cancer in animal cells is twofold. Alright, the virus can actually carry genes that directly cause cancer. This is going to be an oncogene. Or the virus itself produces or allows for the production of proteins that induce the loss of growth regulation. So a virus infects a host cell and it carries with it genes, and those genes are going to downregulate or inhibit the cell to act normal. So some major cancer-causing viruses, papillomaviruses, alright, that's a big one. 80 to 90% of cervical cancers are caused by papillomaviruses, and that is because the physician scientists do a biopsy of that cervical tissue, and they can find the virus there. Epstein-Barr virus is a herpes virus that causes a lot of different cancers. And then HTLV is a T-cell lymphoma virus that can cause T cells to go rogue and become cancerous. And that is the end of chapter 5.3, looking at the life cycle of viruses and also their ability to cause cancer.